Bismillah ar-Rahman ar-Rahim. This is 5090 O-Level Biology Syllabus 2023. Chapter 5 Enzymes is the second video in which we're going to discuss the practicals that we're going to conduct on the effect of temperature and pH on the rate of enzyme activity. Now, the syllabus is very clear that we're going to discuss this point, investigate and describe the effects of temperature and pH on enzyme activity. So whenever there is an investigate, it means we've got to know the practical procedures. Now we start with the first experiment, which is how are we going to study the rate of enzyme activity. So we're going to study the rate of enzyme activity, but what are we going to study it? How does temperature affect it? So the first thing which I always tell you is that you've got to take 5 temperatures. If I ask you to study the effect of pH, then you will take 5 pH. So if I say study the effect of temperature, then we're going to take 5 temperatures. Now I've given you two options. Either you take 25, 30, 35, 40, 45, or you take 50, 60 and 70. Now this is for you to decide which 5 temperatures are you going to take, but you have to be careful about certain things. I'm just going to discuss that with you. You've been given a 1% starch, you've been given 5% amylase solution, and you've been given iodine solution. So you got three things. One is the 1% starch, 5% amylase, and how do you make this 1% starch, by the way? You take 1 gram and you add water, and then you mix it up, and you then add enough water to make it 100 ml, right? You take 1 gram and you add water, and then you add enough water to make the total solution 100 ml. So 1 out of 100, 1 gram in 100 will be 1%, 5 gram in 100 will be 5%. Amylase is in the form of a powder. Just like if you've ever made pizza, the pizza comes in sort of these granules in a little tin in which you add it to the flour, and then you knead the flour, and then you make the roti for the pizza. So you have to study the effect of temperature on the rate of enzyme activity. Now, there's something I want to explain to you. It's not given in your book. If you look at this graph, the optimum temperature in this graph is somewhere here. This is the optimum temperature. Now, just imagine if you took 5 temperatures, which were 10, 20, 30, 40, or 5, 10, or 10, 15, 20, 25, and 30. Now, do you realize where the mistake was going to be? You were only going to get readings here, 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 and here. So you would just get a graph like this. But if you took more temperatures, then you would get a graph like this in which it would decrease. So if you took temperatures which were less than the optimum, then you would get a graph just like this. You'd only get a partial graph. And then what will be your conclusion? Your conclusion would be increasing temperature increases the rate of enzyme activity. But that's actually wrong, because you only took this temperature, this, and this. So you took 5 temperatures which were below the optimum. So this you have to be careful, is that you have to take temperatures which will cover the optimum as well, but then you don't know what is the optimum. So if it's a human enzyme, yes, then the optimum is 37. But if it's some other enzyme, then the optimum would be different. So this is what you have to understand, is how do you decide, and when do you decide, and what sort of temperatures should you take. So you should take temperatures which are in a sort of, they should be between say 25 and 55, because roughly most enzymes in plants and animals work best between these range. Of course, there are some enzymes which work best at even 100, which we've talked in the previous video. Now, how do you test for starch? Now you can see this is the iodine solution. That's the color that you see in iodine. It's yellow or brown. And if starch is added to it, then the food sample, it turns this black color. So this is the test for starch, and that is why we've given you iodine solution with the amylase and the starch. Now, what is the basic rule first of all? Let's try to understand that. First thing first, you have to keep the amylase 1 centimeter cube. So you must specify the volume. Whatever the percentage of the amylase was given to you, you were taking 1 centimeter cube of that. So the volume has to be specified. Then you took 5 centimeter starch. But look at this, look at the important thing. The amylase was kept separate and the starch was kept separate, and then this was kept in a water bath. So this is a water bath, and say the water bath was at 30 degrees Celsius. So now the amylase is also at 30 degrees Celsius and the starch is also at 30 degrees Celsius, and you leave this for 5 minutes and then you mix them. This is what you've done. You mix them now and you maintain the temperature at 30 degrees Celsius. So starch and the amylase mixture, and you've done that after 5 minutes, and now you put the stopwatch on, and now after every 30 seconds, what are you going to do? You're going to take a sample of this and you're going to mix it with the iodine solution. And what have you got? You've got a spotting tile with iodine in the wells, or you can just have a plain tile and you can put iodine on it. And you take this sample, you take a sample from here and you mix it with the iodine solution, and it turns black. If it turns black, it means starch is still present. Then after another 30 seconds, you again touch and it's black. Another 30 seconds is black, another 30 seconds is black, another 30 seconds is black, and now when you put it, it's brown. So you realize 30 plus 30 plus 30 plus 30, so 1 minute, 2 minutes, so it took 30 into 6, 180 seconds to digest all the starch. I'm going to repeat myself. You take the 1 centimeter amylase and the 5 centimeter starch and you keep them separate. Then you mix them and you keep them at 30 degrees Celsius, and then you take a drop and you mix it with the iodine and see what is the color. The color is black. So that means the starch is still remaining. There's still starch left. It's not all of it has not been digested. And then after another 30 seconds you take and then you mix it and it is blue black. And then you mix it again and it is blue black, and then again after 30 seconds. So 30 seconds, 30 seconds. And then last time that it was brown here. So it took 180 seconds to digest all the starch until all the starch has been digested. So this will be the rate of enzyme activity. Please remember, rate is always time. So this was the rate of enzyme activity. Now you will repeat this experiment at the other 4 temperatures, whatever temperatures you've chosen, whether you've chosen 30, 35, 40, 45, and 50. So you will repeat these at the other temperatures. And this is how then you will plot a graph and see the effect of temperature on the rate of enzyme activity. Now we're going to study the effect of pH on the rate of enzyme activity. So we take 5 test tubes and we add 2 cm cube. Please remember, 2 cm cube volume must be decided. 2 cm cube of 1% starch solution in all of them. But now we are not going to add the amylase. I'm not going to add the amylase yet. Amylase that we have, I've decided I'm going to use 1% amylase and I'm going to use 2 centimeter cube of it. Yes, I've decided the volume and the percentage. Now I'm not going to add the amylase, but what I'm going to add is I'm going to somehow alter the pH of this. Now do you see how I have altered the pH? Now another very important thing for you all to realize is that the total volume must remain the same. Now it's written in your books as slightly wrong. I don't agree with that. What is given in the book, it's scientifically wrong. So now this is a total volume of 2 centimeter plus 1 centimeter. So it's 3 centimeter. Now when I add amylase to it, now when I add 2 centimeter of amylase, right? So 2 centimeter of amylase. So now it is 2 plus 1 plus 2. So it's a total of 5 centimeter cube in each test tube. So the total volume must remain the same. So it's 2 centimeter cube of amylase which I have added, which I have colored green. Now all the test tubes are same. Now I'm not saying you do this all in one go. You will have to do one test tube at a time. But I've given you the overall appearance of it, how this is going to do. You can't do this all in one time. You have to do one at a time. So the first test tube, when you mix the amylase, now you take a tile, a white tile, and you add iodine drops to it, right? And then you take a drop of this mixture. So I'm going to make this yellow, the drop. And then you're going to take this drop with the help of a pipette, and then you're going to mix it with this, and you're going to see the color, and the color is black. So starch is still present in it. Now you will do this after every 30 seconds. And you take another drop from here, and then you mix it. It's black, 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 black. Now you realize it's black, and now it's brown. So it has taken 30 into 7, 210 seconds for all the starch to be digested. So you did this with the first test tube. Now you will mix the amylase, because I've just shown you the amylase, but I didn't mean that you should mix the amylase. So as soon as you mix the amylase, then you start doing this on the second test tube. Then when you've done that and you figured it out, how many seconds did it take, then you do it with this test tube, and then you do it with this and this. So 5 pH and the rate of enzyme activity has been studied by altering the pH. Now, what have we kept constant? We've kept the starch solution, which is the substrate constant, the volume, and the concentration. I said the volume and the concentration has been kept constant. So the number of active sites are constant. So if 1% amylase in 2 centimeter has 100 active sites, so all of these have 100 active sites. Even this has 100 active sites, this, all of them have 100 active sites. And in 2 centimeter of starch solution, we have 1000 starch molecules. So all of these have 1000 starch molecules. So you've kept the starch molecules constant and the active sites constant, and what have you changed is the pH. And then you plot a graph and see how the rate of enzyme activity is affected by pH. Now this is a typical graph of effect of temperature and pH. So this is the temperature graph and this is the pH graph. Now you realize if you had taken only 5, 6, 7 pH, then you would only get graphs like this. You'd only get a graph like this. But if you took 5, 6, 7, 8, and 9 pH, then you would get a graph like this. So this is how you have to realize that you must take a few pH less than the optimum and a few more than the optimum. So optimum pH, denaturing of the enzyme, and extremes of pH in temperature, increasing enzyme activity increases the till the optimum, and then after the optimum there is less effective collisions and the rate decreases, and final denaturation is at this point. All of the enzymes, all the active sites have lost their shape, all the enzymes have been denatured. A very interesting graph showing representing the average time it takes for starch to disappear in seconds in relation to the temperature. And you can see this is the temperature graph and the average time for starch to disappear. As we are increasing the temperature, it is taking more time for the starch to disappear. So it's a very interesting graph, and I think sometime maybe Cambridge can use it in an MCQ. Here is a typical reading of pH of starch solution and time taken for the amylase to break it down. So you can see how at 4 pH it took 110 seconds, at 5 it took 50, at 6 it took 20, and then 7 it took 40, and then at 8 it took 70 seconds. But you see, now what we have to realize is it has to be between, it is somewhere around 6. It could be between 5 and 6, it could be between 6 and 7. So then we have to again do this experiment to between, so we must take 5.5 and 6.5 to see whether, if at 6.5 it took 10 seconds, so that means then the peak would be like that. So this graph is something like this. So 4 pH, 5 pH, 6 pH is somewhere here, and 7 pH is somewhere here. We don't know this point. So then we would have to take more values between 5 and 6 and 7. Now I want you to understand that we're going to use living cells, either potatoes or liver cells. And what they contain is an enzyme called catalase. And that breaks down hydrogen peroxide to water and oxygen. So it creates a vigorous reaction and a lot of foam is generated. So this is how it helps you to find out how fast the reaction is. Now we're going to extract and test an enzyme from living cells. And the enzyme to be extracted and tested is catalase, and the substrate is hydrogen peroxide. Certain reactions in the cell produce hydrogen peroxide, which is poisonous, and catalase makes the hydrogen peroxide harmless by breaking it down to water and oxygen. Right? Now, what you do, you grind 1 gram of liver with 20 centimeter cube of water, and you will, when you grind it, actually it's going to release the liver cell content, so which contains the enzyme catalase. So you filter the mixture and you add to test tube A and test tube B. And then you take hydrogen peroxide, and then you take this filtrate and you add it to the hydrogen peroxide, and it will form this foam on it because you are producing the oxygen. The oxygen is going to be released. So you grind a small piece with water and then you filter it, and then you filter the mixture and share between two test tubes A and B. And the filtrate will contain a great variety of substances dissolved out from the cytoplasm of the liver cells, including many enzymes. And because enzymes are specific, however, only one of these catalase will act on the hydrogen peroxide. So what you do is you add this, as I just said, you add this and you add it to the hydrogen peroxide, and there's a lot of fizzing, and the oxygen can be tested with a glowing splint. Now in B, you're going to boil it. So you boil this filtrate. You boiled the filtrate which contained catalase. And now you will add this boiled filtrate to hydrogen peroxide, and there will be no reaction. So there will be no oxygen produced. No reaction, no oxygen produced. Now, what does this show? It shows that that filtrate contained an enzyme, because enzymes will be denatured on boiling. But if you take manganese oxide powder in a test tube with some water and pour this into some hydrogen peroxide, this will also give you a vigorous reaction similar to the one with the liver extract. But if you boil the manganese oxide with water, but if you then take this manganese oxide and it is if you boil it and add it, it will still produce a reaction. The reason is manganese oxide is a catalyst. Manganese oxide is a catalyst, but it's not an enzyme because heating has not altered its catalytic properties. This experiment can be repeated with a piece of potato to compare the catalase content with that of the liver. And the potato, of course, should be the same mass. As I said, grind 1 gram of liver. So the next thing you can take is 1 gram of potato and then grind it, and then similarly filter it and do the same process again. So basically, this is extracting and testing an enzyme from living cells. And hydrogen peroxide is a substrate, and the enzyme which we have extracted from the living cells is catalase. This finishes this video. In the end, I would just like to say a little dua for everybody. ربنا آتنا في الدنيا حسنة وفي الآخرة حسنة وقنا عذاب النار. Our Lord, give us in this world that which is good, and in the hereafter that which is good, and protect us from the punishment of the fire. Thank you for watching and thank you for subscribing. This completes the chapter on enzymes, and then we will start the next chapter in the coming videos. And this is now the syllabus which I'm following is the 2023 new O-level biology syllabus. Thank you once again.